In this video, we will discuss about how the blockade of neutrophil that are recruited to the tumor cells can improve the efficiency of checkpoint blockade immunotherapy. So what happens in normal case is that the neutrophil are recruited towards the tumor cells, and then it can reduce the immune efficiency in a way that it can suppress the immune function towards the tumor cells by interacting with the checkpoint inhibitors on the immune cells and deactivate the immune cells. We will discuss these things in brief in this video. So what is the main idea of this paper? Is to block this neutrophil recruitment to the tumor cells so that the immune system can work efficiently on the tumor cells. So how they do that? They do that by by injection of this by injection of this complex. That is, this complex, they called it as this BCS SAPC complex. So what this complex does is, this complex, when it is injected into the mice along with this PD-L1 antibodies, the PD-L1 antibodies will prevent the inhibition because of the receptors, the PD-L1 receptors that are present on the immune cells. By blocking these PD-L1 receptors, so this so the combination of this PD-L1 antibody and this BCS SAPC is injected into the mice. So the function of this BCS SAPC complex is to prevent the neutrophil recruitment to the tumor cells. And how that does that? It is shown here that BCS SAPC complex interact with the neutrophil, which are in a way towards recruitment to the at the site of tumor cells. And when it's interacted when it is interacted with the this PCS SAPC is interacted with this neutrophil, it prevents the intravasation of this neutrophil at the site of infection. So this inhibit the recruitment of this neutrophil at the tumor site. And so because of this, what will happen is that this neutrophils has this PD-L1 receptors. It will interact with the this PD-L1 ligand on the that are present on the T cells, and thereby inhibit this T activated T cells function. And the other way these neutrophils inhibit this T cells function is by releasing these immunosuppressive cytokines, and this will also deactivate the function of T cells. So by inhibit the recruitment of these neutrophils at the site of tumors, these two ways of the neutrophils to inhibit the immune functions will be stopped or will be reduced to a greater extent. The other way by which they can improve this immune function against the tumor cells is using this PD-L1 antibodies. So these PD-L1 antibodies, if this PD-L1 antibodies is not present, what happens is that these tumor cells will also has some of these PD-L1 receptors. So the tumor cells can interact with the tumor cells can interact with this PD-L1 ligand that are present on the immune cells, and they can deactivate or suppress the immune response against the tumor cells. However, when these PD-L1 antibodies are injected into the mice, so these PD-L1 antibodies will interact with these T cells infection. So because of this, what will happen is that the PD-L1 ligand will already be blocked by these PD-L1 antibodies, and the CD8 T cells or T cells in general will not be deactivated by the PD-L1 receptors that are available on the tumor cells. So these T cells will continue to function. Uh, the T cells will continue to perform its immune function against the tumor cells, and its activity will not be suppressed by either by the tumor cells, PD-L1 receptors present on the tumor cells, or either by the PD-L1 that are present on the neutrophils, as as the neutrophils recruitment is already been inhibited by this BCS SAPC complex interaction. So in this way, by blocking the neutrophils and also these PD-L1 receptors on the immune cells, uh, the efficiency of the immune system will be improved to a greater extent. And in this paper, they can see the uh, life expectancy of these mice that are in which this both both this BCS SAPC complex and PD-L1 antibodies injected is high compared to the control case, where there is no injection of both of this where there is no injection of BCS SAPC complex or PD-L1 antibody complex. So uh, if you need to so this provide us a pathway to somehow work towards preventing this neutrophil recruitment at the site of tumors in the patients, in the human beings. That will also provide a positive results towards these immune functions against the against the tumor. If you need to understand more about this, if you need to understand more about this approach, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.